Well, welcome to the 8 to Tim's Take, and today we have a very special guest, Dr. Dale Blevins. He's a plant physiologist, plant nutritionist, and I can't believe he's been retired for 14 years. But Dale, I worked for you for 14 years.
Oh yeah.
So that's kind of a milestone, and it took me until I was age 35 to be able to call you by your first name because I respected you so much. And finally, you told me to call me Dale.
Yeah.
So I'll probably call him by his first name, but that's not out of disrespect. That's just I feel so comfortable around him.
Sure.
So Dale, you were very lucky in that you were raised in Ozark, Missouri, on a farm.
Yes.
And then you got to come back to Missouri to serve.
I feel really, really lucky about that because I sort of traveled around to get educated, and like a lot of people. My first real job was at the University of Maryland in the botany department, and I was there for 3.5 years. And it was really important for my career because at Maryland, as soon as I got there, I was 1 mile from Beltsville, USDA. Mm-hmm. They had like 100 physiologists there.
Oh, wow.
And they said, okay, you're going to be the secretary-treasurer and then the vice president and the president of the Washington section of the American Society of Plant Physiology.
Oh, wow.
So they had a plan for me because I was the new guy at Maryland. And it turns out that's the biggest section of the Plant Physiology Society. So I got to meet all of these pretty famous people in plant physiology. And then like the Smithsonian closed their radiation biology group there in Washington, D.C., and they went to NASA, some of them, the younger ones, and they really helped me, and some of them went to USDA competitive grants when it started, so that really helped.
Sure.
So I knew a lot of people, and it really helped my career early on, I think.
That's so important, isn't it, to get that. And before we get going too far, what is plant physiology? Because a lot of people don't know that term. Okay.
Well, there was a little book from a guy from Stanford, and I got it a long time ago, and it says, plant physiology is the physics and chemistry of how plants make a living.
Oh, wow. Yeah. Yeah.
So it's like what plants do to grow and make a living. It really is. That's how you can say it simply that way.
And, Bill Spallin, one of our colleagues, he's always said, what makes plants tick?
Yeah, exactly.
All of that.
It's kind of understanding plants. And
yeah,
I mean, even though I grew up on a farm, a lot of times, in our lab, we ground up plants
right.
a lot.
Yes.
You kind of look what's inside and what makes them tick.
That's right. And you mentioned growing up on the farm, but you were also part of that Sputnik generation. You always came back to that.
I'm glad you mentioned that because that had a big influence on my life because at that time, Southwest Missouri was second to Wisconsin in dairy.
Oh, and you're a dairy farmer.
And I grew up on a dairy farm, and all the farms around, even though some people just milk 3 or 4 cows, but everybody had a dairy, even no matter how small. And my neighbor had 200 golden Guernseys. They had a big farm. But there was a kid there just a year older than me. Neither one of us took VOAG because Sputnik came, and we were both good students, and you will go to college.
Yeah. Right.
It was just different at that time.
It was the sciences, because you mentioned plant physiology, physics, and chemistry. That becomes so important.
Yeah.
And you actually majored in chemistry, didn't you?
I was a chemistry major as a BS degree, which I think was really important because plant physiology is the physics and chemistry of how plants make a living.
Right.
We already talked about that.
And how many times we would stare at the periodic table to try and figure out what we were seeing.
Exactly. And I still do that. Yeah.
First thing I did when I took over Sanborn Field was buy a periodic table to put on the wall.
Yeah.
Because that's what we're really talking about.
I still use it all the time.
Right.
Yeah.
Because it's amazing. You can see why plants are reacting or what just by that.
And you see potassium over there on the left, and you see rubidium under it, which is a good isotope for potassium because there's not a good potassium isotope.
Right. So. But they compete too.
Yeah, they compete.
Yeah. So you can look at that. So there's so much.
A lot of kids take lithium tablets because they have potassium activated enzymes that are out of control. So you give them lithium, and that slows them down.
Oh wow, yeah, because you can solve a lot of issues that way.
Yeah.
So when you came to Mizzou, then after 3.5 years at Maryland, you had a great group starting by that time, didn't you?
Oh well. Number one, a friend of mine, Doug Randall, was already here in plant biochemistry, and he was thinking about forming a plant group. And now we call the IPG, and we've had like over 40 years of having the IPG. It has like 55 plant people in it, plant physiologists
from all walks,
all kinds of plant biotechnologists and stuff.
And it's funny on that. Now you see. The students are coming back and being featured speakers.
Oh yeah,
that we had last time.
Yeah. Oh yeah, exactly.
But here you worked on uriides. That was like you were really getting up on soybean nodulation. And uriides. And
so when I came, I wanted to. When I came to Missouri in '78, I wanted to work on soybeans, and we were just learning about urea, nitrogen, and I'd worked with Harold Evans, who was a famous guy to work with, and he was working on nitrogen fixation, so I had an interest in that. Mm-hmm. And then about that time, Tetsuo Matsumoto in Japan had discovered that soybean nodules are different than what we'd learned in all the textbooks about what's transported from the nodule after they fix nitrogen. The form of nitrogen is not amides, but it's something called uriides, which we didn't even know what it was. But uriides are real simple. It's like a sandwich with urea being the bread and acetate being the meat inside. So that's all a uriide is. But it's 4 carbons and 4 nitrogens. So soybeans are very efficient at getting that fixed nitrogen up to the leaves and up to the developing pods.
See, that's really interesting. So red clover is going to be different, isn't it? It's going to have a different
clover. Yeah, there's cool season legumes, and they put asparagine upstairs, and uriides are warm season legumes, and they put uriides upstairs, and they're more efficient. They're more efficient at translocation,
and that really helps explain some things because in my career so far, I've noticed that soybeans are great in fixers compared to some of our cool season.
Absolutely, yeah.
And now with our soybean yields getting pushed so high, they can't keep up.
Yeah.
So we kind of have to rethink all of this now.
Yeah, hopefully they'll be more interested in nitrogen fixation. There's never been quite, in my view, enough attention sustained in nitrogen fixation to keep it going.
We took it for granted that it's there.
Yeah.
But I think now when we expect more out of that, we're going to have to revisit that, aren't we?
Yeah.
And maybe a lot of the research that you all did, that's something to build on a different way, because the basic research is what is a framework for these future discoveries.
Yeah. I think we have Gary Stacy's lab now at Mizzou. And he's a big nitrogen fixer and has been his whole career, but he's about the only one left that we have there.
I've talked to some of the other physiologists and breeders and wondering, have we unintentionally selected for high N2 fixation for soybeans, for higher yields? Because that becomes a limiting factor.
Oh yeah.
That we just maybe they repartition more down there to the nodules or something.
Yeah, it is an expensive process for the plant, but it takes a lot of sucrose going down there to feed it.
Right.
For energy, yeah.
But because y'all had ones where you knocked that gene out, and so it didn't fix nitrogen.
Yeah.
And yields were a little higher.
Yeah.
if you fed them enough nitrogen.
If you fed them artificial nitrogen,
But really,
fertilizers nitrogen.
But really, in today when we're trying to reduce our petroleum input, because it takes a lot of petroleum to make synthetic nitrogen.
Yeah,
We want that biological fixed nitrogen.
exactly. But it's not a giveaway really, because it's expensive for the plant to do it too. So,
But it's getting that from sunlight.
yeah, it's sunlight.
Yes.
It's got sunlight.
Right. So there's give and take.
Solar power.
So understanding these principles, and that's one of the things that you always taught, getting the mechanism down. If you can get the
Yeah.
mechanism figured out, then you can go from there.
Then you can tweak it.
That's right. Because you always
Yeah.
preach that
Yeah.
mechanisms.
Yeah.
Getting them, and you all pretty well got that figured out with the ureides and such.
Right. Yeah.
Hi, Dale mentioned the work that he did with ureides. Well, soybeans and other legumes, as you know, have a symbiotic relationship with bacteria. And what we have done is that we have a oversized nodule grown here, and they're usually quite much smaller than that compared to the roots, and I have many of those. But what we have is that we have into nitrogen, that's just free nitrogen that diffused from the atmosphere into the soil, and the plant can't use that. But these certain bacteria can that are, in soybeans case, are called rhizobacteria, and they infect the root, and as a result the root will grow this nodule or this mass around it. So inside then we have the bacteria, and it can take this atmospheric nitrogen that the plant can't use, but it can reduce it into a form the plant can use. And in soybeans, we call those ureides. Now, in other legumes like clovers and alfalfa, it's a slightly different compound, and asparagine is what it is. But these ureides then are a form of nitrogen. They're carbon and nitrogen put together that can be transported then by the plant up into the leaves. So then we see it start transporting up to the plant. It can go into the leaves. It can go into the developing pods. But this is how soybeans get their nitrogen, from that symbiotic relationship between the soil bacteria, the Bradyrhizobia, and the plant. Now this is a carbon sink for the plant, and some of the data that we found was that when we had supernodulation, where we had 10 times more nodules on the plants than we normally have, we actually reduced yield because it took so much carbon from the plant to help feed the bacteria. But this is what we're working on for the last 40 years, trying to get other crops like corn or wheat actually have a relationship like this. And so far our scientists have not gotten that far, because there's a complex gene interaction between the bacteria and the plant that has to be overcome, and also all of these different pathways. So Dale worked on this a lot with other scientists here at MU about how ureide metabolism is important. It has nickel as a big cofactor into all of this. So it's just not a very straightforward process. It's very complex, but it really gave us an understanding of how we can improve the nitrogen content of our soybeans.
Now, something else that you and Doug and others that were in, I got to thinking, I'd hate to start naming names, I might forget somebody, like Dave Emmerich and Joe Polacco.
Yeah.
You had quite a few folks working on this.
Oh yeah. We had a real group. It was a group effort at first,
Right.
and then we for a while we had, and you were in on this. We had Roy Morris and
Yeah.
We didn't have any hormone people.
Mhm.
Then we got Tom and Gretchen from Minnesota and Roy Morris from Oregon State, and suddenly we had oxen people and cytokinin people.
Putting all of this
And-
together.
and they all came together. Yeah.
Right. But one thing that you all really led the way in for the rest of the college university was getting competitive grants,
Oh yeah.
federal, because people didn't know how to do that.
Yeah. That's where my experience at Maryland came in, because I sort of, when I was teaching, we used to have kids write papers, and then I got to thinking right off the bat that they shouldn't be necessarily writing papers. They should be learning how to write grant proposals. So I required grant proposals in my classes, in my graduate classes.
I remember doing one of those. That's hard
because it's tough. But writing grants is tough. But like when I started my career, I didn't know much about grant proposals. So,
really.
So writing,
Mm-hmm.
yeah. I guess because I'd worked with Harold Evans, I got a grant about the first year at Maryland, so that really helped my career.
Sure.
Get me started. And I thought, well, my grad students in my classes ought to start doing that.
Because if they're going to succeed in their career, they're going to have to be able to write grant proposals.
Right. Yeah.
And
that's a fact of life.
It is
science.
And your students came out knowing how to do that.
Yeah.
Whether they were your direct students or your indirect students.
Yeah.
Ones you taught in class knew how to do that. And I still remember mine from that class. And some of it was, I can say it wasn't the best idea, but others I thought was pretty good, it's a pretty good idea.
Yeah.
You mentioned cytokinins. We did some studies with Roy Morris and cytokinins.
IVs.
Yeah. And I ran across some of that data the other day, and I was looking at it. I thought, we were so far ahead of everybody that we couldn't get this published because we were really way ahead of everybody else.
Yeah.
So that's always neat. But let's talk about your teaching, and we'll go back to research here in just a little bit. But you mentioned teaching.
Yes.
You taught a very different way. You talked about the grant proposals, but you taught your graduate classes a little differently, didn't you?
Well, yeah, I tried. I learned early on that it seems like a lot of professors on graduate classes, they were having students give talks, so they weren't really teaching much.
Yeah,
in my opinion. But so my idea was to have students learn how to read papers, and really critically. And so, I sort of got in trouble with one of my grad students at the time because I was collecting. There was this one guy at a university that we all know about, but I thought he was dry labbing his research.
Ooh.
Bad. So I still have the file of the papers he pushed. He was working on uriides, so I knew about uriides, and I knew how hard it was. But he was publishing. In other words, his wife was a biochemist, and she died. So at first he was probably okay, but then he just kept publishing.
Well, I know exactly who we are now.
Yeah. But anyway, I would have students learn how to read papers, try to teach them how to read papers, how to break them down. And then they would get pretty good at it. And then I would throw one of his papers in. And they would get so critical.
That's good.
It was funny. But
early on, they wouldn't have been critical.
Yeah, they wouldn't have been critical.
They would say, oh, this is great.
Yeah.
They won't ask me to review papers anymore.
Really?
I'm pretty critical.
Yeah.
It's got to be done right.
Oh, it's got to. Yeah, and it's a hard job, So
to do it right, I mean, you can rubber stamp something, but if you really want to dig down into there.
But it's important because when you publish your journals, two or three people around the world have to review those. Mm-hmm. So it's good if they've had some experience,
right,
doing that kind of stuff,
critiquing them so they know how to do it better. And I think as you taught a lot of folks, I'm thinking like Jeff Valenick and with the Purdue and others, they carried that way of teaching to their students.
Yeah.
And that's really a very popular way of teaching still today.
Yeah.
Graduate school, type classes that you review papers, you learn from those papers and then discuss it. Now, one time though, teaching class actually got into switched your career as far as to a whole different section on boron, just because you were doing the
Oh yeah.
your mineral nutrition class and the. Well, I'll let you tell it.
Well,
That's one of my favorite stories.
well, this is a this is kind of my career. So when I went to Maryland, pretty famous plant nutritionist there who had written a book. He retired, so he got to pick me out of the line, out of the 50 applicants or something. And he was working on boron. So when I left Oregon State, Harold Evans, he was so highly respected, he said, "Now, if I were you, I would not work on boron until you get tenure." Because at that time, we didn't know anything about boron. But so I probably broke Hugh Gow's heart when I went to Maryland, and I didn't work on boron. I started working on nitrogen and stuff. So anyway, boron is a hard subject because we know it's required, but we don't know much about why
Right.
it is. And there, at that time, early in my career, there's like 20 hypotheses, and it was so confusing. I sort of quit. I would just tell students boron's required. We don't really know why, and that's all. I didn't have a lecture on it.
Right. But one time you were writing the nutrient deficiency symptoms of, let's see, toxicity of aluminum. You just finished that.
I see that story. Yeah.
And then you started on boron. What did you notice?
Yeah. Then I noticed there were similarities there. So, and actually, that's what I've been thinking about lately. I'm getting ready to write a opinion paper on boron and see. I think boron, it is needed for it. We know in the pectin parts of cell walls. Other than that, we still don't know too much about it. But I think the big deal is we have this toxic aluminum in acid soils all around the world, and manganese in some soils are both, are very available in acidic soils. And so I think boron attacks those two elements and keeps them under control. That's not a real classical requirement for plants, but it sure helps plants grow in these acid soils with high aluminum or high manganese.
And you showed that.
I've showed it.
Right.
I've shown it, but I don't think enough people know about that, or we don't have enough information on that. So that's why I'm writing this opinion paper.
Yeah.
And I think I'm going to be attacked around the world.
Well, I actually was giving a talk one time, probably to an international audience, and I pulled out that classic photograph that you have that shows how the alfalfa roots grew through the high aluminum soil.
Oh yeah,
with when you added boron.
Yeah,
and they hadn't seen that before.
No, I don't.
That is,
and, and we have a couple of papers on that, but it's hardly cited.
Yeah. Because what you saw in your class, aluminum toxicity and boron deficiency are very similar.
Very similar, yeah.
So there's a relationship, and that really set the stage. And again, it's one of those things. The research is there. It may not be accepted today, but down the road, it could be a huge factor.
Yeah.
Because one of the things you made us do, like when we write a paper, and we cite a citation, you don't go to that person may have cited that a previous paper. You go all the way back to the original paper.
Exactly.
Cause sometimes it's misinterpreted too.
Exactly. And I don't see enough of that now when I read papers that going back to the original discovery.
Right. Because that's really important.
Yeah.
It's because you're taking somebody else's opinion on it, but you need to go back to the person that really did that.
Especially as you get older, you appreciate that.
Well, we found out that a lot of Germans in the 1930s, they did a lot of work.
Oh yeah.
They had some wonderful plant physiology work. That you could cite them, and they did it.
One of my favorite original discoveries, which came right off the farm, was how deeply rooted plants in a drought can bring up water for little shallow rooted plant companions. I thought that was such a wonderful, brilliant, original idea. So Steve Korak came over from Maryland to work here, and we were talking about that problem. He decided to do it, and he had the audacity to go to the library that very night, and he found that in 1930, in where was it? It was in New Mexico, I think. That this guy had written an Ag Experiment Station bulletin showing that very thing.
Oh.
And he wrote two Ag Experiment Stations showing deeply rooted plants can bring up water. But we still went ahead and did that. He hadn't published it. I think he was an extension guy, so he hadn't published in a referee journal article. So we did it, and
yeah, and you did a little bit more of a enclosed environment in the greenhouse.
We did a really big study.
Where you could really to make sure that was it. I remember that, and then you followed that up with Jeff Hobbins' nutrient transfer.
Yeah, like potassium can transfer from deeply rooted plants to shallow rooted plants. So but there again we had to use rubidium 86 for potassium. But so,
that was one of my first jobs with you, was to take that experiment down. I mean, it was done.
Oh yeah.
I think we redid it and just get the final, and so that was really neat. I learned a lot about that.
Yeah.
But because and now you think about all we talk about a lot is having a lot of diversity out there.
Yeah.
In our pastures and
exactly
whatnot. Doing the same thing. That's why we have it.
So can I tell you the story about how I got onto that deeply rooted plant spring?
Yeah.
Because when I was really young and way before you were born, like in, what year was it? 52 to 56, there was a drought, and I was on the farm, and everything was brown. Our lawn, we were raking grasshoppers up and burning them. It was so dry, So we were driving around, but we had alfalfa fields, and as alfalfa fields got older, we would sprinkle orchard grass in there to fill out the holes, and so we had alfalfa orchard grass. So, and then in pastures we had all orchard grass. It was before fescue got popular. So that was fescue. Orchard grass fields were all brown, but the orchard grass growing with alfalfa was green. And that's where I got the idea that alfalfa is deeply rooted. Noted, it's one of the most deeply rooted plants that we grow. But so it must have been bringing up water for its companion orchard grass. So that's where that experiment came from. All those years passed before.
Wow.
Steve Korak did the experiment.
But observations on the farm.
Yeah.
And that's another thing, even though you may be doing lab type equipment studies, you still need to get out and look.
Oh yeah.
Observe what's going on out there. You can learn a lot.
Yeah, exactly.
Like this morning we were doing bulk densities out in the field in different rotations and length of rotations, and you can learn a lot just getting out there in those fields and learning so much.